before I became a director of this institution, I was invited to many international and national conferences for my research work, the area of my interest and my capabilities. But after I became director of CCMA for the last 5-6 years, I get invited to conferences which have nothing to do. I have no expertise in it, but I still get invited to go and give some talks. So this is one of those. I am not a cardiologist and really don't work in any area related to heart, except that part of my research, part of molecule that I work with, happens to be related to cardiomyopathy. That is one link that I have. But in general, of course, I was trained as a chemist, but slowly moved into biology, molecular biology, and medicine. And as just now I was being introduced, my major recognitions and achievements are in the medical area. So I've received the Bhatnagar Award in medical science and Ranbaxy in medical science. So I had a journey from chemistry to medical sciences, and I enjoyed this. Heart diseases are increasing all over the world, perhaps largely due to lifestyle changes, because human being is not designed to work like this, eat like this. Our body, through the evolution, is supposed to be hunting, gathering, working, and eating sufficiently. But now we eat and sit. So since this is the change in our lifestyle, this lifestyle change is causing the major problems, including diabetes and cardiovascular diseases. In addition to lifestyle changes, genetic predisposition is one of the major aspects. So world over, scientists are looking at genetic aspects and molecular biology basis for certain diseases. CYP2C19 is the one which is required to pre-process the drug, then only it will work. So the person who has a mutation in here, there will be no use. This drug won't work if the person is a mutant for this, and it can cause problems. So you have to choose another drug. Now, whether going through genetics, looking for a patient gene and looking for a SNPs, can we decide the course of therapy is the question that we got asked. It looks like that it is possible to do it, but there are a lot of experimental issues. One is that we must know that is one-to-one correlation. It is not the probability. It must be very clear correlation. And second thing is ability to sequence. How many hospitals can actually take a patient's blood and take DNA and isolate and sequence and then identify the mutation? That's a very big challenge. Today, technology is changing, and we may go further in these technology developments. For example, long time ago, when DNA was sequenced, it was sequenced by a method for chemical methods, which was designed by Maxim and Gilbert. People who have studied MBBS long time ago, you must have remembered that the Maxim-Gilbert method of sequencing DNA, which is impossible to sequence. It takes about day or two to sequence about 10 to 15 nucleotides. Then Sanger method came in, where the chain termination approach was utilized, but still we have a gel and 4 lanes and things like that. Then that is replaced with the same Sanger method, but by fluorescence labeled method with a capillary method, instrument method. Then we could sequence 650 to 700 nucleotides in 1 or 2 hours time. So it is a very fast, very good method. But still that is not good enough for us, because we are talking about 3.15 billion nucleotides. So it is not possible to sequence it that way. Then the next generation sequencing came in. The next generation sequencing, we don't use separation methods. We use imaging methods. We have an array on it. You build the molecule by synthesis, and you take a photographs. All photographs you combine, and you will be able to generate sequencing information. Massively parallel, synthesis-based sequencing approach. That is what we call next generation sequencing. With the next generation sequencing, we can sequence human genome in 2 to 3 days' time. I'm sure you remember, in 2000 when human genome sequence was announced, it was about 3 years effort by 6 countries. But now we can sequence human genome in about 4 to 5 days time with the advanced technology. Even that is not sufficient for a clinical level testing. Now the technology is further enhanced. We have a next generation sequencing technology where we don't even synthesize. We take the DNA and pull it through a hole. For example, like a needle. Take a needle, eye of the needle, put your thread, pull from one side, you will sequence it. That's it. Take a DNA and sequence it. How do we do that? When DNA is getting through this hole, DNA has A, T, G, C, different nucleotides. each nucleotide has electron density differences. When it is going through, there is a current differences or capacitance difference will be different for each nucleotide. So pull through and measure the pulses. Each pulse will tell you what is the sequence it is. This is ion core technology, which is a new technology, which has been already tested, and it will become available sooner. Through one hole, one can pull the DNA and one will be able to sequence it. This may take about a couple of hours to do the process. But knowing Japanese capabilities, Japanese will put 1,000 such holes on one chip, and so we'll make 1,000th of 2 hours. So that is the possibility of sequencing. So we may see in our maybe 2 to 3 years' time, human genome can be sequenced in 15 minutes' time in a clinical setup. So if that can be seen, then you will be able to prescribe the medicines based on pharmacogenomics. Whether this drug will work for the person or what drug should be chosen is possible to do this. So this kind of a massive technological development is taking place, and I hope that this will be greatly useful for treatment decisions.